Start by imagining a big city. In the city, we have power plants that provide all the energy needed to run the entire city. When these power plants break down, the whole infrastructure in the city will collapse. Now, can you imagine the same things happening inside the cells in our body? The power plants of our cells are called mitochondria. Like the power plants, they are responsible for producing most of the energy that the cells need to be functional. So if these mitochondria breaks down, the whole infrastructure of the cells will collapse, just like in the city. But when this happens inside the cells, it can lead to the development of different neurological and cancer diseases, depending on which cell type it happens in. So this means that this signaling, how does that happen? When the mitochondria become dysfunctional, they will send a signal to the nucleus, which acts as the control center of the cells. Depending on which signal that the nucleus receives from the mitochondria, it can change the gene expression, leading to either a healthy or a diseased cell. This means that this signaling road from the mitochondria to the nucleus can potentially be used as a new target for treating cancer and neurological diseases. But we have one main problem here. We don't know which molecules are involved in this signaling road. Like in a big city, we have a lot of traffic inside the natural cells from all the different compartments. So it can be really hard to identify the specific signaling road from the mitochondria to the nuclei. So what I aim to do is that I want to create a minimal artificial cells where I can study this direct signaling road without having the rest of the traffic from the natural cells. So what I did was that I isolated the mitochondria, being the power plants, isolated the nuclei, being the control center, and encapsulated them into a gel, create these tiny minimal cells containing only nuclei and mitochondria. And in that process, I discovered three main findings. One, I could isolate the nuclei, and they could keep their main function of producing RNA for up to 4 hours. Two, I could isolate the mitochondria, and they could even produce energy for up to 2 weeks. Three, I could measure a change in the gene expression level indicating a healthy or a diseased cells. This means in a minimal model, we can not only study how a healthy city runs, but we can also study what happens if the power plant starts breaking down. So with that, we take one step closer to understand the role of dysfunctional mitochondria and how they can lead to different diseases. Thank you.